All right. So the next speaker is going to be Yue Zhang, and he's from the Paul G. Allen School from University of Washington, and he's going to be talking about scalable preprocessing of sparse RC single-cell RNA-seq data exploiting prior knowledge.
Okay. Hello. Hi, so my name is Yue, and I'm a grad student in Georg Selig's group at the University of Washington, and I will be presenting on our paper on UNCURL, which is a method for analyzing single-cell RNA-seq data. So I'm sure a lot of you are already familiar with single-cell RNA-seq data. Basically, as compared to previous bulk experiments, we capture each cell individually and sequence the mRNA in each cell individually. This allows us to perform a lot of new kinds of analyses, but also leads to a lot of new challenges. Single-cell RNA-seq can get us potentially a lot more information than bulk RNA-seq data. By sequencing single cells, we can get a sense of the heterogeneity of the population. We can classify cell types or perform lineage analysis, which lets us understand the differentiation processes and developmental processes of cells. But single-cell RNA-seq also presents a number of new challenges compared to bulk sequencing. The data sets are generally much sparser and have more noise. This is due to the effects of the sampling process. We don't actually sequence every single mRNA molecule present in the cell, but instead we sequence some random sample of that cell, and that random sample might be a lot sparser. The method that we have created for dealing with this problem is called UNCURL. It doesn't actually stand for anything. So it takes as input the observed gene expression matrix, which is the matrix of genes by cells, and it outputs a true transcriptomic state, which is a product of two low-rank matrices, M and W. I'll describe what they are in the next slide. So the basis of our model is something called the convex mixture model. The model has two key assumptions. The first assumption, described by the equation here in the figure on the right, is that the true gene expression is a convex mixture of archetypes. Basically, each cell lies somewhere along a low-dimensional simplex, where the corners of the simplex are defined by M, which is a matrix in gene expression space, and the position of each cell within that simplex is defined by W, which is a matrix of weights that sum up to 1. As shown on the figure on the right, cells can be either belong to one state, in which case it's at the corner of the simplex, or it can be a mixture of several states, in which case it's at the edge of the simplex. The second part of our assumption is that there is a noisy channel going between the true gene expression state and the observed gene expression state through single-cell RNA-seq. This is a noisy channel that is parameterized by a sampling distribution, which is parameterized by the true gene expression state and possibly some other parameters. To actually use our model, we need to know which sampling distribution we want to use. In our implementation, we have three candidate distributions, the Gaussian, log normal, and Poisson, but we also can use many other distributions, such as the negative binomial or zero-inflated distributions. We have a heuristic method for selecting the distribution we use. First, we fit each distribution to every gene using maximum likelihood. After that, we calculate the fit error, which is just the distance between the empirical or observed distribution and the estimated distribution. And the predicted distribution is the distribution that has the lowest fit error across all the genes. In this example, the predicted distribution is the Gaussian. When practiced, we usually use the Poisson distribution for count or UMI value data and the log normal distribution for RNA-seq data that has already been transformed through something like FEKM or other normalization techniques. So now to optimization. Like many other machine learning problems, our algorithm works by finding the maximum likelihood solution in this probabilistic model. This is a matrix factorization problem, which we solve using alternating minimization. We first hold M constant, solve for W, then hold W constant, solve for M, and iterate until a certain number of iterations have been reached or we reach convergence. The actual optimization algorithm used is different for the different distributions. In the case of the Gaussian or log normal distributions, our problem reduces to NMF, or non-negative matrix factorization, after some transformations. This is a very well-studied problem in machine learning, and there are many pretty fast algorithms for this. In the case of the Poisson distribution, we use the no-Lipschitz algorithm, which is for non-Lipschitz optimization, to solve an alternating Poisson regression problem. And the runtime of our algorithm is proportional to the number of non-zero entries in the data matrix times k, which is the latent dimensionality or the number of cell states. So unlike a lot of other algorithms, our algorithm actually takes advantage of the sparsity of the data to improve our performance in inference. So onto performance. This shows the performance comparison on two different data sets, one small data set and one very large data set. On both data sets, UNCURL performs a lot faster than other methods, both methods specialized for single-cell RNA-seq and methods that are more general like t-SNE. It also consumes less memory because it doesn't need to compute like, say, something like a distance matrix between the cells. UNCURL is also scalable through parallelization. As we have more processors in our computer, we can throw more processors at UNCURL to make it run faster, I guess. Applications. UNCURL can be used for many different purposes. It can be used as a general purpose pre-processing tool in conjunction with other downstream tools for tasks such as clustering, visualization, lineage estimation, or interactive analysis, or it can be used as a tool for clustering on its own. It can also take into account prior information to improve the performance of the inference. The first task that we'll describe is clustering, which is a very common task for single-cell RNA-seq analysis. UNCURL already gives us a soft clustering through the W matrix, which, as we mentioned before, is a matrix of the weights or the locations of the cells in the simplex. We get a hard clustering from that just by taking the argmax of each column. We compared UNCURL's performance in clustering with many different methods, including both generalized dimensionality and clustering, dimensionality reduction and clustering algorithms like t-SNE and PCA followed by k-means, as well as specialized algorithms for single-cell RNA-seq. And on this data set, which is the TOSIC 2016 data set with 49 mouse brain cell types, UNCURL performed better than all of the other algorithms. And we tested both UNCURL using the method described earlier or UNCURL with other clustering algorithms running on top of the output of UNCURL. And similar results can be seen for many other data sets, which are in the paper. One of the advantages of UNCURL is that it can use prior knowledge to improve inference. We can use prior knowledge in many forms, as listed on the right here. We can use gene expression information, such as from bulk RNA-seq data or microarrays, or qualitative information, such as lists of marker genes. One problem with using these kinds of information is assay incompatibility. For example, with bulk RNA-seq or microarrays, the distribution of the genes could be very different from the distribution in single-cell RNA-seq. And that's also a problem for qualitative data. To deal with this problem, we use a method called qual-norm, which transforms the prior information, which can be either qualitative or quantitative, into a form that can be used for UNCURL. The reasons that we want to convert the prior information into an initialization is because UNCURL is a non-convex algorithm, so information in the initialization will also be present in the outcome. This is usually a bad thing, but in this case, we can turn it into a good thing, basically. The first step in using prior information is to binarize it. First, we take the median of each gene, and then the values above the median are set to 1, and the values below the median are set to 0. This is based on the assumption that gene expression is usually either on or off in a binary state. After that, we work on the observed data, not the prior data. On the observed data, we cluster each gene's cell count, gene's count distribution, into two clusters, one high cluster and one low cluster. Next, on the binarized prior information, we replace all the values that have a value of 1 with the high cluster center and the values that have a 0 with the low cluster center. After we do that for every gene in the prior information, we now have a matrix that can be used to initialize UNCURL for W. So results for, we tested qual-norm on the Z-cell dataset, which is a brain cell dataset. This dataset contained a list of annotated cell types, and there was another dataset that had bulk RNA-seq data for some of these cell types. So the graph on the left shows dimensionality reduction with UNCURL using various initializations. The unsupervised is a purely unsupervised initialization. Bulk on the bottom left whilst using the bulk RNA-seq data directly as initialization. aggregate is sort of the cheating method that uses the actual cluster means from the single cell data set, and qual-norm is the method we described earlier. Just from a qualitative perspective, qual-norm seems to improve performance compared to unsupervised and bulk. On the right, we showed how the NMI changes as the number of genes we use for semi-supervision increase for these different methods. Qual-norm generally performs better than using bulk semi-supervision. Another thing that we can use, task that we can use UNCURL for is lineage. It's not used for lineage estimation directly, but in conjunction with other algorithms. First, we run UNCURL, then we run the other algorithms, such as monocle2 and slicer here, on top of the output of UNCURL. One advantage of this is that UNCURL can be used in a semi-supervised fashion to take into account prior information before doing the lineage analysis. And as shown on the pictures here, generally UNCURL has a higher rank correlation with the true ordering of the cells than just running these methods on the data directly. Finally, we developed a tool which we called UNCURL-App, which is a tool to help biologists analyze single-cell RNA-seq data. This has several goals. First of which is just to create a web-based UI for UNCURL to allow people to use UNCURL without programming. And also, we want to simplify and streamline how biologists perform single-cell RNA-seq analysis in a way that also incorporates prior knowledge from both the user and external sources. This is the URL here. This is very preliminary work, as you can tell by the URL. Here we define a pipeline for cluster analysis using one of the data sets that came out of our lab. This process was inspired by some of the work that was done in our lab in analyzing this data set, which was a very painstaking manual process that involved a lot of data-dependent steps. The first step is in our interactive analysis pipeline using UNCURL-App is to run UNCURL on the data, which gives us a clustering and a visualization. This is a screenshot of UNCURL-App. On the left, we see a interactive view of the cluster of the cells colored by cluster. On the right, we see a view of the top differentially expressed genes for a given cluster. After that, we can try to identify the biological relevance of the cell types to understand what all this actually means. This shows the expression levels of one gene in the data set. This is the clustering entropy, which is a metric that comes from the W matrix of UNCURL. It's just the entropy of every column. A higher value indicates more uncertainty in the clustering labels assigned by UNCURL. We can also integrate external data sets. On the left-hand side, we see the results from running Enricher, which is a gene set enrichment analysis library on the gene set for one specific cell type. After this, the user can then interactively work with the analysis by either merging or splitting a cluster. And after merging or splitting a cluster, the entire analysis pipeline is rerun, giving us a new visualization, clustering, and differential gene expression. And this allows the user to basically have an iterative pipeline through the analysis. In conclusion, basically, UNCURL is a method for analyzing single-cell RNA-seq data by matrix factorization, and this output can be used for various tasks such as clustering, lineage estimation, and dimensionality reduction. And each of these can involve semi-supervision. And our code is open source for both the app and UNCURL itself is on GitHub. And for future directions, there are a number of things we want to improve with UNCURL. The first one is how to identify K, the latent dimensionality, or the number of cell states, without having to manually specify it. We can do this using the interactive analysis, or we can try to have some sort of like non-parametric models. We also want to avoid having to specify a sampling distribution. So instead of like saying the Poisson or log normal distribution or whatever, we just learn a noisy channel directly from the data. We can do that using something like variational autoencoders or other neural network approaches. And we also want to incorporate outside information in like both more and different kinds of information and incorporate it in a better way. We might want to move to like incorporating literature information or information from like databases of bulk RNA-seq experiments. And finally, thanks to my advisor, thanks to the funding sources, thanks to Paul G. Allen, and special thanks to Sumit Mukherjee, my co-author, who put a lot of work into this and who unfortunately couldn't be here today. Yeah. Well,
Hey, very nice work. I don't think it's on. Hello? Yes. I guess I'm about the sampling distribution. A priori, I'd sort of expect, right, that a negative binomial, a zero-inflated negative binomial, might be the most appropriate one.
yeah.
So two related questions. One is, if you did use a zero-inflated negative binomial, what impact would that have on your running time? And then also, have you evaluated, even for the ones that you did mention, empirically, which ones capture the data most best?
Well, based on empirically, for UMI valued or data sets, the Poisson distribution generally works the best. But even better than like negative binomial distributions often have some like computational issues, which is part of the reason we didn't include it.
Yeah, that's why I asked about running time.
It was much, we don't have like a no-lips kind of algorithm for the negative binomial yet at least. You might have one in the future.
So some of the stuff that you were talking about reminded me a little bit of archetypal analysis.
Yeah.
So did you compare it to archetypal analysis, which we actually used a lot in the magic paper, for example? From the Urialan lab.
No, we did not.
So that they use PCA-based convex hull method
Yeah.
to solve some of your intrinsic dimensionality problems.
Okay, thanks.
Yeah, I just wanted to follow up on the negative binomial question. I
Yeah.
admittedly have skin in the game, having played around with it for a while. But going back to the early days of bulk RNA-seq, it was shown pretty convincingly that the sampling variation for RNA-seq data is Poisson.
Yeah.
And so any source of variation at all above sampling variation gives you an over-dispersed Poisson model.
Yeah.
And so a negative binomial is a natural fit. So
Yeah.
I can understand wanting to use a Poisson for convenience or computational convenience,
Yeah.
but I guess I need to see more to be persuaded that it's actually capturing the variation that we see in the data. And have you done simulation or other sort of experiments to show that these models that are computationally convenient are also actually capturing the relevant variation in the data?
That's a good question. We have done some simulation experiments, but I think with the negative, even if it's data sample from the negative binomial distribution, the Poisson distribution still captures a lot of the variance, like the variation present in the data set that's useful for downstream analyses. But like, yeah.